Okay, so hello everyone. Basically, so what's going on is that I think that I didn't find the Embryology 4 lecture very helpful, so I just wanted to see if I could try to explain it in a little bit different way. Here we go. Basically, what this lecture is about is how the embryo gets its orientation, body axes in the embryo, right? So it's a question of the embryo just starts out as a blob of cells. How does it figure out which way is up, which way is down, which side of the embryo is going to be the head, which side is going to be the tail, which side's left, which side's right? And this is important because even though the outside of a human body might look symmetrical, the visceral organs are very much asymmetrical. The heart, as most people know, is on the left, but also the liver should be on the right side, the spleen should be on the left, the sigmoid colon should be on the left. The large intestine's asymmetrical. The appendix is going to be on the right. And it's important for these to be in the correct locations. And if they're not, then it could lead to problems. So let's see how the embryo figures out which side is which. Basically, in this lecture, we're going to go over the genes and the genes that drive the process of figuring out which side is which, and also the cellular, what it looks like at a cellular level as the embryo works out, it gets its orientation. So first let's look at this is gastrulation, right? So this is what the embryo looked like at the end of week 3. We have gastrulations happened, and we have the cranial side, the caudal side, the left side, the right side. So this blue layer is the ectoderm. The yellow layer is the endoderm. And the red layer is the mesoderm. I probably shouldn't write in there, the mesoderm. So if we look at it from the dorsal side, if we look at it from the dorsal side, we'll just see the ectoderm. So that's what we're looking at here. And here we can see the primitive node and also the primitive streak down here. And looking at it, you can already tell that the primitive streak is on the tail side of the embryo, and the primitive node is on the cranial side of the embryo. However, the rest of the embryo doesn't quite know this, and in order for it to figure it out, we have to have some very specific genes being expressed. Specifically, over here in this region right here, in the anterior visceral endoderm. Okay, so what does that mean? The endoderm, obviously it's endoderm, it's yellow, it's on the ventral side. They call it visceral. I don't know, I'm gonna pretend that is like that's a synonym for ventral. And it's anterior because it is on the cranial side. It's on the anterior side of the embryos. I guess cranial is technically superior once you get to the once you become a person, but for now it looks very much like this is the anterior, the front side of the embryo. So I will allow this terminology. So how does this anterior visceral endoderm let the embryo know that, hey, this side is cranial, the other side is caudal? Well, what it does is it will inhibit nodal. So there's this protein. Let me go to purple. So there's a protein called nodal, I'm going to use N, and it's going to be secreted from the primitive node, which is, roughly right here. That color. Flat. Okay, it's going to be really hard to... Okay, I'm going to just get myself a nice blob going so I can find this color later easily. Anyway, so... there's a primitive node, and also behind the primitive node, I should say caudal to the primitive node, we have the primitive streak. So nodal is going to be expressed all throughout here. So really wish I had the, come on. Oh wait, can I do that? Okay, that's fine. That's fine by me. Okay, no, take away that tool. All right, so sorry about that. But yeah, so we have nodal protein here, and let's say that nodal I just realized that this is close to the primitive streak color, so we're going to change that to black again. So here's the primitive streak, there's the primitive node, and there's nodal, right? So nodal is going to be expressed throughout the caudal side of the embryo. So we're gonna have it there, but on the anterior side, we're not gonna have any nodal. Why not? Because this anterior visceral endoderm, it's gonna secrete some proteins, specifically Cerberus and Lefty1, that inhibit nodal. So these guys are gonna go, they're gonna say, no, we don't want any nodal being expressed on the anterior side. The node needs to stay in the middle and not, and why is it called nodal? Because, you know, it's expressed in the primitive node and posterior to that, but not beyond the primitive node. And why won't it go beyond? Because Cerberus and Lefty1. How do I remember, how can you remember these protein names? So Cerberus, remember, is the three-headed bear. So it makes sense, the three-headed dog, sorry. Cerberus is the three-headed dog, and it makes sense that it would be expressed on the head side of the embryo, on the cranial side of the embryo. Lefty1 is generally pretty important, and we'll see it later, so hopefully it'll stick. Dr. Rylon also talks about 3 other proteins that are expressed by the anterior visceral endoderm. Those are OTX2, LIM1, and HESX1, but I think these 3 are pretty low yield because they don't show up like ever again. So I think the main ones to learn are going to be Cerberus and Lefty1. So what is this? The anterior visceral endoderm, it makes Cerberus because it's on the head side, and also Lefty1, and these stop nodal from spreading. To the head side, and because they do that, that's what allows this side of the embryo to form into the head. If you delete Cerberus, what actually happens is that the organism just does not have a head, because nodal takes over here, and then there's no space for the head to form, because a head, as far as the embryo is concerned, is a place without nodal. And on the other hand, if you overexpress Cerberus, what happens is that the nodal gets inhibited even over here. Nodal gets too inhibited, and there are too many heads forming, which is why it's called Cerberus. You can get like a 3-headed, you can get a multi-headed embryo that way. Anyway, so yeah, so let's move on though. So basically Cerberus and Lefty1 can inhibit nodal, and now we have a head side. Okay, what else? We also have, but that's not all, right? Because that's never all. So there's more to it. In addition to this, we also have, if you remember, there are some cells that they're epiblast or ectoderm cells that are migrating towards the primitive streak. And, you know, as they come into the streak, they're going to go and they're going to become the mesoderm or endoderm. Let's focus specifically on the ones that go on to become mesoderm. Of those cells, some of them are supposed to form this notochord structure on the cranial side. And that notochord structure is important for the neural plate development and development of the nervous system. And so without that, we won't have a nervous system. So again, this is also going to, you know, what's an important part of the nervous system? The brain. So this is also going to be related to head development. So this is another important part of how the embryo knows, like, which side is the head and which side is the tail. So, oh, right, I forgot I can do that. Oh no, the angle's off. I don't know how to fix it. I'm sorry. Oh, I think I did. Okay. Anyway, so let's just leave it there. So here's what's going on, right? So we've got, so the caudal, right? So I was saying, how how are we going to get this notochord here, right? So the notochord is going to be made of mesoderm, but specifically it's going to be mesoderm that stays on the dorsal side. So some of the mesoderm, it's going to fall all the way to the ventral side, and some of it's going to stick more on the dorsal side of the embryo, sticking closer to the ectoderm. And so what are we going to do with that? So okay, so the important thing is that there's some genes that need to be expressed for the notochord to form properly. So specifically, there is this gene, I'm going to use orange for this one, there's this gene that's, at first at least, it's expressed throughout the embryo. And this gene, what it does is it's called BMP4. And I call it the belly-making protein. That is not what it stands for, but it helps me understand what's going on. It tells the mesoderm cells to fall all the way to the ventral side. So if there's a cell that comes in through the primitive streak and then comes around here, then if it runs into BMP4, then it will fall all the way to the bottom, and it'll settle on the ventral side, and it'll be making basically the ventral surface, if you remember, is the belly of the grown adult. So these make belly mesoderms. But what if we want to make the notochord? We need to stop this. We need to stop this protein from doing its thing. We need to stop belly making protein. We need to stop the orange. And the way that's going to happen is that we can stop it with. It has a couple of inhibitors. There is, first off, there's the cordon, because it's going to allow, remember, this is going to allow the notochord to form. There's also noggin, because the notochord is going to form on the cranial side, and your noggin is like your head. And finally we have follistatin. Now, I don't actually know why they call it follistatin, but I'm going to pretend it's because it's going to put a stasis on the mesoderm cells that are falling onto the ventral side, right? So it's going to stop the mesoderm cells from falling to the ventral side. So it's going to be stopping the fall. And that's how I'm going to remember it. You can come up with a different mnemonic if you want, or you could look up why they actually call it follistatin, but I don't care that much. So here we go. We have these three proteins that are going to stop the belly-making protein and allow us to make our notochord. So these proteins, where are they going to be expressed? Again, these are also going to be expressed in the primitive node. So these are going to be expressed in the node over here. So in that node zone, we're going to have cordon, noggin, and follistatin. And so they'll basically, when these are expressed, they will allow us to form the notochord. How will these be activated? Well, first off, in order for these to be activated, so we do want to make some belly, we do want to make some ventral mesoderm initially, but once we're ready to make the notochord, what we'll do is we'll activate a protein called goosecoid, which then activates cordon, which will inhibit the BMP4. So if we inhibit the BMP4, we will erase all this BMP4, particularly on the cranial side. So goosecoid and cordon, their gradient's going to be more like, it's going to affect the cells that are in the node and cranial. So instead of nodal, whereas nodal was expressed like from the node and towards the tail end, the cordon will be affecting cells that are in the node towards the head end. So this is where cordon works. So goosecoid will activate cordon, and that'll stop BMP4. So we'll erase all of the BMP4 on the head side from the node forward, but the ones in the back will stay. So now that we've done that, what happens is that we can have our cells, our mesoderm cells, can go and make the notochord. Okay, great. Now we've got a notochord. And if you remember, notochord stimulates the development of the neural plate up here. Once the notochord develops, then we can have the neural plate, and now we can have that neural plate. So let's go over this. Pathway one more time. So there's goosecoid will activate cordon, and along with noggin and follistatin, cordon will inhibit BMP4, which then allows us, once BMP4 is gone, then we can have the notochord forming. Okay, and so the formation of the notochord, they call it dorsalization of the mesoderm, because then now the mesoderm is going to stay on the dorsal side. And before we inhibit it, we have ventralization of the mesoderm, because the mesoderm is going to be falling onto the ventral side. So there's that. We talked about like where these are expressed, like location-wise. And yeah, that's it. And then there's one more, right? So there's one more gene that's important here in terms of differentiating cranial and caudal. It doesn't really fit in with either of the pathways, sort of, that we've talked about so far. So this gene is what we'll call, let's go for a different color here. This gene is going to be expressed basically this is another one that's going to be expressed from the node down. It's called brachyury, also known as T. I don't know what the T in parentheses is, but whenever they write brachyury, they always write T in parentheses. Anyway, point is that this is a very important gene. They mentioned it last in the embryo 3 as well. This gene basically lets the embryo know to make the dorsal side. So it's going to be expressed throughout. Sorry, not the dorsal side, the caudal side, the tail end of the bottom half of a person. If you don't have brachyury, you will not have a bottom half, because this is the master gene for making lower limbs. So without brachyury, you have they have some pictures on the side. You can go through the PowerPoint. I do recommend going through the PowerPoint just to make sure that you get anything that I missed. But basically, the person does not have legs. That's it. Someone who has a deficient brachyury will not have legs. So yeah, big very important thing. It basically lets you. It tells the embryo that hey, we are on the caudal. Tells all the cells on the caudal side that they are on the caudal side, and that they need to differentiate accordingly into leg cells. Without brachyury, that doesn't happen. Okay, so that's pretty much it, right? So let's do a review. Okay, I don't like the fact that it can rotate. I just want to zoom. But anyway, so let's do a review of the important things. Let's pick a different color so we can highlight. I don't know, like this. Okay, so important proteins, right? How do we know that we're on the head side? Well, there's this important tissue over here, the anterior visceral endoderm, that will make Cerberus and lefty 1. These inhibit nodal, and what is the definition of a head? A head is a place where there is no nodal. So then that's how we know that we have the head. How do we know that we are on the tail side? We have brachyury. And another thing that we need in order to make heads is we also need to know that we also need this BMP4 to be off, so that we can make a notochord, so we can get our so we can make the the spinal cord and everything. And where does that come from? Well, when we're ready to start dorsalization of the mesoderm to make the notochord, we'll turn on goosecoid, which will then turn on cordon, which then turns off BMP4, which allows the notochord to form. So I guess in order to make a head, you have to have Cerberus and lefty 1 working, and you also have to have goosecoid and this whole pathway thing working as well. And in order to make a tail, all we need is brachyury, as far as I can tell here. Nodal probably also plays some role, but yeah, there's that. And so let's talk about disorders. So if you have overexpression of goosecoid, let's look at this, right? So overexpression of goosecoid, we'll use pink. Will it show up? It will. Overexpression of goosecoid, right? What will that present as? You'll overexpress cordon, So you will have very, very low BMP4. So you will erase the BMP4 in places where you shouldn't have erased it, which allows you to make basically, you get dorsalization in places where it shouldn't have happened. So you might get like maybe even 2 notochords forming, right? So we might even have a second notochord start to form over here if we if we just erase all of our BMP, then this would allow too much of the mesoderm to dorsalize and become a notochord. And so what this presents as is you get a person with 2 heads. So 2 notochords, you could get 2 heads. So you can see it in this slide. This is one cause of having multiple heads. Another one, as we talked about, would be overexpression of Cerberus, because then it gets rid of your nodal in places where nodal shouldn't be removed. If you remove BMP4 where in places where it shouldn't be removed, extra head. If you remove nodal in places where it shouldn't be removed, again, extra head. If you and we also talked about, I think, if the primitive streak doesn't form properly, that can also cause an extra head. So there are a couple of pathways where messing up will result in a 2-headed person, and I'm honestly surprised that 2-headed individuals aren't more common after these last 2 lectures. But anyway, so there's that, and yeah, so there's okay. So that's what we've got for the formation of head to tail. And I'm gonna take a break here and save this video. Okay, so now we'll take a look at how the embryo tells the difference between left and right side. So again, here we have the, this is again a view from the dorsal surface, so we're only looking at the ectoderm. Yeah, so here we can see the primitive node, and here we can see the primitive streak. And if you remember from last lecture, one thing that's going to be important here is we have, there is a protein that is called FGF8, and that is going to be secreted here. Actually, I'll go into it later. But there's a protein called FGF8, fibroblast growth factor 8, that is expressed here in the primitive streak. Right, so this is going to come in. This is going to be important in a minute here. So let's remember that. So how can the embryo tell the difference between left and right? So what happens, basically the core of the mechanism comes down to cilia. There are cilia in the node cells, and they beat from the right. They beat from the right to the left. And then they kick back, they bend back, and then they beat right to left. So this is like they have a certain direction of movement. So because those cilia are beating right to left here in the primitive node, they're going to be creating a little current of they're going to be creating a little current of, like, the I guess it's amniotic fluid on this side. And what this current's going to do is it's going to lead to a slight accumulation of protein called nodal 8. Sorry, just nodal. Actually, we already talked about nodal. And yeah, so that's the same nodal from before. This is the purple one that I was doing before. So it's going to lead to an accumulation of nodal over here on the left side. So there's nodal. And on the right side, we're going to have instead, we'll have an accumulation of, let's go with that burgundy color or whatever, or I guess it's just brown. Accumulation of monoamine oxidase, MAO. So monoamine oxidase, interesting. Have you ever heard of MAO before? I think we actually mentioned it in, Dr. Reddy's mentioned it in this class. So MAO, it's monoamine oxidase is one of the enzymes that degrades epinephrine. It stops epinephrine signals. But here it's going to be interesting because it also degrades, besides epinephrine, it will also degrade other monoamines such as norepinephrine, dopamine, and serotonin. Serotonin is going to be particularly important for us here, so I'm going to make it red because it's so important here. 5-HT is the abbreviation for serotonin. It's like 5-hydroxy something, something with a T. Serotonin is going to be expressed initially everywhere throughout the ectoderm or throughout the embryo. But what we're going to do, what happens is that the monoamine oxidase is going to selectively degrade the ones on the right side. So because of that current that was created by the cilia, we ended up with monoamine oxidase accumulating on the right side and nodal accumulating on the left. And the monoamine oxidase that was left on the right side, it degraded all of the serotonin on the right, leaving serotonin only on the left side. So now we have serotonin on the left side. What's that going to do? What the serotonin does is it will further activate FGF8. And FGF8, what it does is it will increase the expression, or let me use the color schemes here. So 5-HT will activate fibroblast growth factor 8, which was originally in the primitive streak, but it's going to enhance that secretion, and what that's going to do is FGF8 will enhance our secretion of nodal. So FGF8 it's going to enhance the secretion of nodal, but also the secretion of lefty 2. So last time we saw lefty 1, this time it's lefty 2, and I guess I will actually remind y'all of what lefty 1 was doing, because it's helpful to keep that in mind, for me at least. Lefty 1 we saw over here. There's lefty 1. Lefty 1 was being secreted in the head region by this anterior visceral endoderm to basically let us know that, hey, this is where the head is. So lefty 2 is also involved in the same area of signaling, but it does something slightly different. This is going to be, the lefty 2 is going to be more important in determining which side is left and right. Although lefty 1, it will have a role in right-left expression as well. So let's go there and see what happens. So first off, lefty 2. What lefty 2 does is it will then activate. It will then activate this very important gene called PITX2. This is the master transcription factor for left-sidedness. So, like, I guess it probably stimulates the development of the heart, the spleen, whatever else is on the left side of the body, but not on the right side, all that stuff. So this is like the master transcription factor for left-sidedness. So, and I somewhere, she's not very clear about how the cascades work in the lecture, but somehow nodal also leads to enhanced secretion of PITX2. But ultimately, the point is that serotonin, FGF8, nodal, lefty 2, all of these are going to be increasing PITX2, which is the master transcription factor for left-sidedness. Okay, so there's that. So now we have a question that we have to answer, and that is why, sure, so the serotonin, it gets degraded by monoamine oxidase if it tries to cross over to the right side. But why not, why can't say PITX2 cross over to the right side? Or why can't nodal cross over to the right side, right? So the nodal, because that's being expressed here, why can't it just hop over and go there? It's not being degraded, is it? Well, it's not, but there is, if you remember, there's a notochord up here, right? So this notochord is going to be going this way, and the notochord, what it does is, for its part, it will be releasing SHH, Sonic Hedgehog, named after the video game character. We theoretically learned about it in biology. I still don't know what it actually does, though. but SHH, Sonic Hedgehog gene, is going to be in the notochord, and the Sonic Hedgehog, as well as another, the lefty 1 from before. If you remember, lefty 1 was being expressed in the head region, so I guess maybe Sonic Hedgehog where the notochord is, and lefty 1 more over on the cranial side. Lefty 1 is going, and Sonic Hedgehog, these guys will prevent nodal from crossing over to the other side. So they'll inhibit nodal expression on the right side. So then that way we only see nodal on the left, and we only see PIDX on the left here. So there's that. All right, so yeah, so that's where everything is, like, spatially. And also, let's just take another look at this, right? So where do SHH and lefty 1 come in if we're talking about like this? So lefty 1 and SHH are both going to be inhibiting nodal, specifically on the right side. So they're located in like the center, and they're going to block expression of nodal from spreading over to the right, crossing over to the right side. All right, so that's, now we have laterality, right? So we've got the left side, and we have this side that is not left, but what genes are there that tell the right side to be the right side? Like, I mean, someone's got to tell the liver to go on the right side, the liver, the gallbladder, etc. And pretty much the only gene that they mentioned is snail. It's not very well studied, so I guess snail is the only transcription factor they know about on the right side. And yeah, snail's on the right side. I think that's all you have to know. Cool. So that's left and right side. Okay, so all right, she does a fine job of explaining like the clinical stuff here, but I'll review it anyway. So what happens if you screw up any of these genes, right? So there's a reason they call these lefty 1 and lefty 2, because if you screw it up, then the left side is going to be abnormal. So what would you get? You could get a disease called situs inversus. This is if the whole body is flipped left and right. So we have a person, and so if their heart is on this is their right side. If their heart is on their right side and their liver is on their left, that would be situs in, if all of the internal organs are flipped left and right, then that's called situs inversus. The normal version, if your organs are normal, then that would be called situs solitus. I'm not pronouncing any of these correctly. And then if you only have one organ flipped, so like let's say that your heart is in the wrong side, but your liver is fine and everything else is fine. And your gallbladder, your spleen, everything's fine, but just your heart's on the wrong side. That's called heterotaxy. Hetero is like different, right? So it's different because one of the organs is on a different side from all of its usual friends. Like the left-sided organs are separated from their friends. They're on different sides, and the right-sided or at least one of them is. So yeah, so situs solitus is normal. Situs inversus is all wrong. This is just one or a few wrong, and situs solitus is all normal. Okay, and this is, so what we talked about is particularly important. This here, 5-HT, is serotonin, right? And, serotonin is a relatively popular hormone in, I guess, a neurotransmitter hormone in popular culture, because it's also, involved in depression. So what happens if you take a drug that affects your serotonin levels, specifically, say, a SSRI? What will that do if you take that during pregnancy? So an SSRI, what does it do? It increases serotonin, right? So this will increase our levels of serotonin, which increases our level of FGF8, which increases our level of lefty 2, also increases our level of nodal, and you see the cascade, right? So we have too much PIDX2 and too much nodal. So what happens is that this, we could have so much PIDX2 or so much nodal that the nodal might be able to overcome the barrier posed by the sonic hedgehog gene, and cross over to the right side and cause some left-sided organs to develop on the right side. So we could get something like heterotaxy or citizen versus. And usually these come with cardiac defects because I'm assuming because it's like one of the because the heart needs to like pump blood through the aorta, and if that's at a weird angle or in the wrong direction, that can probably cause problems for the heart and lead to it weakening over time. So usually, so we generally want pregnant women to avoid SSRIs during the time of the pregnancy because it could lead to heterotaxy, basically. Okay, and so that's left versus right. Okay, so I will pause again, and then we'll talk about the somites. Okay, so I think the second half of the lecture is a lot easier to understand, but I'm going to try explaining it just to see, I guess basically to help myself understand it better. But I think you could, like, if you don't like what I'm saying, then I think that her explanation actually works for the second half. And really, again, it's the molecular stuff that confuses me most. That for the cellular processes, I feel like I get that makes sense. But it's just all the genes that I think it helps to maybe walk through it a little bit more slowly. Anyway, so here we go. I'll go through the slides that she has because, like I said, for the cellular stuff at least, these slides work fine. But we'll just annotate it as we go to help it make more sense. Okay, so this time we're going to be talking about the development of somites. Somites are an important idea. This is basically the embryological origin of the dermatomes that we saw and the myotomes that we saw before. So in the different parts of the arm, there's like different spinal nerves innervate different regions. Like, for example, we'll test C4 up here, and then like C6 down here, and then C7's the middle finger, C8's the pinky finger. This guy is missing a finger. Oh well. And then like T1 is up here somewhere, etc. Right? So it's like the different spinal nerves are innervating different areas. Somites are basically like the origin of where that comes from. Where does the dermatome come from? Where does the myotome come from? Why do different spinal nerves innervate different areas, and why does the map look the way that it does? So, right, so the beginning of this happens with our gastrulated embryo. If you remember, here's our gastrulated embryo. So at the end of gastrulation, you have your 3 cell layers. There's the blue mesoderm. There is the yellow, blue is ectoderm. Blue is ectoderm. Red, there's the yellow endoderm, and the red mesoderm in the middle. So we have these 3 germ layers at the end of gastrulation, and then what we're going to do at the beginning of the somite formation it starts with this change in the mesoderm. Basically, what's going to happen is that this mesoderm is going to form a couple of bulges, and it's going to form basically 2 bulges and then spread out. Same thing on the other side, and let's look at what that makes. When the mesoderm kind of forms 2 bulges, the first bulge we're going to call the paraxial mesoderm. Para meaning next to, axial, it's next, right next to the body axis. It's probably going to happen a lot. My pinky will jump the screen to a different position just because of how the iPad works. But so paraxial mesoderm is this the first bulge. The second bulge is intermediate mesoderm because it's in between the other 2. And finally, we have the lateral plate mesoderm, which I don't know, doesn't really look like a bulge to me, but basically it's the rest of the mesoderm. And all 3 of these are going to be important in somite development. Well, not quite all 3 of them. I guess the main one for somite development is going to be paraxial mesoderm. This is going to be the one that actually forms somites. The other 2 are going to kind of help regulate and stuff, but the paraxial mesoderm, the one that's right next to the axis, that's the one that's actually going to form somites. Why? Because a lot of the signals for somite formation are actually going to come from the axis, like the neural plate up here and the notochordal process in the middle of the embryo. So that's why the paraxial mesoderm is going to be the one that actually forms somites. Yeah, okay. So here's another view of the embryo that's a little bit more advanced than the one that was in the previous slide. You can see it's kind of folding a little bit more. And again, you can see the paraxial mesoderm. There's the paraxial, intermediate, and lateral plate is like that one right there, that little C-shaped thing on the edge. Okay, so somites. So I think it makes sense to look at this here. Somites are these little bulges right here on either side of the neural tube. So what happened, and the order that these form in is that first you get this cephalicmost one, and then you keep getting them forming in the caudal direction. They start at the cephalic or cranial side, and formation continues towards the tail end of the embryo, towards the posterior end of the embryo. So, and the rate that this happens is about 3 pairs a day in humans. It's different in mice. But yeah, we have about 3 pairs a day in humans, and you can use this to estimate embryo age. So starting on day 20, roughly speaking, you're going to start with about one somite on day 20, and then 3 times like the number of days, I guess. The number of days since day 20 will give you roughly maybe the number of somites on the low end. But it's variable, so I really don't expect them to test us on this. But the idea is that you can count the number of somites and use it to estimate the age of an embryo. But it's just an estimate, so I don't expect to see it on tests. Next, molecular regulation of somite formation. So the molecular regulation, this is the confusing stuff, right? I don't really like this picture. I feel like it's incomplete, so let's look at it slightly different. I have pulled up here. Here it is. So it's pretty much the same picture, but I think it helps to have the second cycle. So here's the idea, right? So first off, you see on the left side in A, where are the somites forming? There they are, the little circles on either side of like the neural tube, on either side of the axis. This is the paraxial mesoderm, remember the red layer that's forming somites. Okay. And what happens, right? So there are 3, can I mark this up? No. There are 3 transcription factors that we need to know that will be expressed at the caudal end. What is caudal? That's like the tail end over here. Remember this is, excuse me guys, I'm writing with an eraser, that's why I can't see it. There's the cranial end over here, and the caudal end over here. And as I said, there's 3 transcription factors that we need to know. On this caudal side, we have FGF8 and WNT3A. The Wnt proteins, the WNT ones, are very, generally speaking, they're pretty important for throughout the process of like embryonic development. So those guys are going to come back. FGF8, y'all hopefully remember that. Remember that was expressed in the primitive streak on the caudal side of the embryo. I guess I'll kind of draw out the diagram from the first part of the lecture. Remember, there's the primitive node, primitive streak, and FGF8 is going to be this thing that is expressed over, remember FGF8 is expressed throughout here, right? I think I used orange for it before, but it's expressed throughout the primitive streak, which is on the caudal side, so that makes sense that FGF8 is expressed over here. And there's also another transcription factor that we need to know called retinoic acid. And unfortunately, I've seen some ambiguous literature, I think, as to whether that's expressed on the cranial side or the caudal side, but it's also going to be involved in this process. So anyway, so we have these three transcription factors, or proteins, signals. These three signals are going to be activating. What all of them do is they start off this notch like wave. So there's a wave of notch expression. So if we check like at a given time, if we check at a given time, there is a what is this? Oh, initially, when FGF8 and Wnt3A and retinoic acid are being expressed in the back, that's going to induce notch throughout this orange zone. But then the orange zone is going to move forward, so the expression of notch here is going to induce the expression of notch here. So now this part is going to be part of the zone, and the expression of notch here is going to induce the expression of notch just cranial to that. So it's going to move. So it's going to move cranially, and after a while, like it gets tired of expressing notch. So that's why you see this blank area over here. So there's like this back end that's also moving forward. So the cells will express notch for a while, and finally, when notch reaches like the very far end, that's going to eventually lead to the formation of the next, the third pair of somites. And so then you see that when notch gets kind of close-ish, then we start another wave, because you could think of it kind of like the action potential. Like, remember the voltage-gated sodium channels are like inhibited for a while, but then after that inhibitory period is over, then they start up, and then they can start up again. So here, same thing here. Notch expression, which is both orange and green, both of those are going to be representing the notch protein. Like this whole box is talking about notch. So orange is this one wave of notch expression, green is another wave of notch expression. So when one wave of notch expression gets close to the cranial end, then another wave is going to start at the caudal end, and again, it's going to propagate forward throughout the embryo, forward through the embryo, until it reaches the front again, and then it's going to induce, this wave is going to induce the formation of the fourth somite. And at the same time, then we'll have another wave of notch starting up at the caudal end. So this is the idea that we have of this clock gene oscillations. Notch is going to be expressed on, and then it's going to be off, and then it's going to be on, and then it's going to be off. So so it's going to be expressed for a while, and then it's going to be inhibited for a while, and that's going to be determining the somite formation. So the important protein to remember for somite formation is notch. That's the big one. You can't, the less important, the slightly lower yield ones are retinoic acid, FGF8, and Wnt3A. Why these ones in particular? FGF8 makes sense because notch expression starts at the caudal end, which is where the primitive streak is, which is what secretes FGF8. Wnt3A is just, the Wnt proteins are generally important, and I guess you just have to kind of memorize that 3A is the one that's involved in notch. And then there's retinoic acid, and I don't really know what he's doing. He's doing his thing. Okay, cool. All right, so that explains the segmentation thingy. What do you call it? The segmentation clock. That's what I was going for. The segmentation clock. Basically, it's going to be notch and Wnt. Oh, and we don't have to know that. Yeah. So notch. The main thing is the cycles in notch, and it's going to be driven by these three proteins here. All right, so there's that. Next, moving on to the somite differentiation. Now let's take a look at what happens at a cellular level again, right? So we're going to take a break from the molecules and talk about cells again. So yeah, I'm gonna swap to my own drawing for this. I will credit my drawing from what's his face, the noted anatomist. I think he does a great video of explaining this. So yeah, you should definitely watch his videos. But what I'm going to try to do is going to take basically his explanation and then add the molecular stuff into that drawing that he had. So here's what we got, right? So basically, I guess I'm going to add a new layer for this. But basically what we've got is, let's remember we had the neural plates. So I guess what we were just talking about is that there was, like, ectoderm, and then we talked about how the mesoderm was kind of forming into an interesting shape, right? And then I guess the ectoderm is, the endoderm is going to be over here. Oof. Yeah, okay, this is very rough, but use your imaginations, kids. There we go. So this is, roughly speaking, these are the 3 germ layers, right? So there's ectoderm in blue, endoderm in yellow, and mesoderm in the pinkish color here. So basically what that's going to, remember, the paraxial mesoderm, we have paraxial mesoderm, we have the intermediate mesoderm, and we have the lateral mesoderm. I don't know if that's right. Let me check that. Lateral plate mesoderm. So those are the 3 here. So what we see is I've drawn below this, I've drawn the process a little bit further along. When it gets a little bit further along, you can see that the 3 mesoderms, I've drawn them separate here. I don't think they're actually, like, completely separate, but I think it helps to clarify, just for clarity, to have some imaginary borders between them. But there's that. There's those guys. And somewhere in here there's also endoderm, but endoderm's not important in somite formation, so we're going to ignore it. The important things, let's talk about what happens to the ectoderm and the mesoderm. Ectoderm, okay? So this was the ectoderm, and it's going to develop into, remember, the skin. The epidermis derives from the ectoderm, so that's why I've drawn it over here. And also the neural tube is going to derive from the endoderm as well, or sorry, ectoderm as well. Ectoderm is going to have the neural tube as well as the epidermis. I guess these neural crest cells, technically speaking, should be colored the same as the ectoderm color as well. Those are definitely ectoderm cells. And I just realized I don't like that shade of blue. I think it's too dark, so we're gonna, oops. I'm gonna swap to a lighter shade of blue for the ectoderm. Anyway, so there we go. And, well, I don't think I'm gonna be able to fix the top one. I'll just go with it. So yeah, there's that. Okay, so now let's talk about what happens to the mesoderm, right? So the paraxial mesoderm, remember, that's the one that's gonna form the somites. That's gonna form the somites. So let me find the right layer, and let's point that out. Paraxial mesoderm is going to become this guy. This is the paraxial. This one's the intermediate, and this one's the lateral plate. So the paraxial mesoderm, I may not have drawn the lateral plate perfectly, but the paraxial mesoderm is going to be the one that's going to form somites. So let's take a look at what happens in that, right? So here we have a cross-section of, like, one somite. So you remember that before we said that there's, like, a neural tube, and you have, like, a bunch of somites forming, right? So if we cut through one of those this way, and then, laid it out and viewed it from the front side, I guess it doesn't really matter which side we view it from, but if we do, like, a transverse cross-section of these somites, this is what we're going to see over here. Okay, I've chosen an interesting layer to draw in because this one's important. Anyway, so I'm going to go ahead and hide that because I think that it's done its job now. So now let's talk about the somite. So the somite, each of these somites is going to be developing into different parts. There's going to be and this is going to happen under the influence of different proteins. So first off, let's talk about what happens, right? So the first thing that's going to happen is that we're going to split into you see that line there? So initially there's no line, but the first thing that happens is going to be the split between let's say this orange part, and man, ooh, I wanted to use the green, right? But I don't have a green here, so let's just wow, legit no green, huh? Oh, I like that. That's a very nice color. Okay, and that green color right there, right? Okay, so what proteins are going to be causing this split? So let's zoom in a little. Let me turn off the bucket. Okay, what proteins are causing this split here? Well, we have, let's see. Okay, so first off, remember, this is, so let's talk about what this is. What is this little tube here? What's this little, what could be a little circle of mesoderm right under the neural tube? Interesting question. Well, so if you remember from last week, we had here's like one view of the, say, of the embryo. And then if you remember, right, so there was like, what happened is that this was, so we have here would be the primitive node, here's the primitive streak, this is the cranial end, this is the caudal end, and if you remember, these cells, they migrated in and they made this little structure called the notochord, right? So here's the notochord under here, the notochord, and that's actually what this guy is. That's the notochord, right? And what else was there? There's also the, oh, and then above the notochord, we got that neural tube to develop. Neural tube. I guess it starts out as a neural plate, and then it folds into the neural tube. It starts folding in the middle, and then the folding spreads cranially and caudally. And then the caudal end, the posterior neural cord, is the last one to close, and if it doesn't close, you get spina bifida. Yeah, that stuff. Okay, so now let's talk about this notochord here. What's important? What proteins are expressed in the notochord? If you remember, I think, yes, one of the proteins that we talked about being expressed in the notochord is the sonic hedgehog gene. SHH is expressed by the notochord. And so that's gonna be important here. And there's another protein that we talked about as well. That's gonna be expressed generally on the head side, called noggin. And we because the noggin is the head. And we talked about noggin along with, I think it was Cerberus and Lefty. Let's actually check to make sure though. Where did noggin come in? So just so we can make these connections. Noggin, because I feel like having connections makes it a lot easier to understand. Okay, yeah, noggin, chordin, and follistatin are all going to be inhibiting BMP4 so that you can form a head on the caudal side. So chordin, noggin, and follistatin, there they are. Those are the three things. Those are three things that are expressed on the cranial side, but not on the caudal side. And those guys, so if we have noggin, which is one of those guys, that's going to be expressed by the neural tube specifically. It's like the ventral part of the neural tube. So what we're going to have is, I probably need a new layer for proteins. Okay, so what we're going to have is the sonic hedgehog coming from the notochord, and noggin coming from the ventral side of the neural tube. Those guys are going to induce this green area to do its thing, and be different from the orange layer. And so if you remember, both of these used to be just paraxial mesoderm, but now the green area is going to become the sclerotome, and the orange area is going to have different proteins acting on it that makes it into something else. So let's write down what the green area becomes. So green is the sclerotome. So the way it works is that noggin from the neural tube, and SHH from the notochord. These guys are both going to activate a transcription factor called PAX1. And we're going to see more. We're going to see another PAX1, a PAX protein in a second. But PAX1, what it does is that's going to be the transcription factor that makes sclerotome do its thing. So that's the pathway there. And then let's also look at another side, right? So how does the orange side know to be orange, and what does that mean? Well, the orange side, it's going to get different proteins. We're going to have Wnt. There's going to be two different Wnt proteins. There's going to be two different Wnt proteins coming from the dorsal neural tube. So I guess the ventral side of the neural tube secretes noggin, but we have the dorsal side of the neural tube. It's going to be secreting something else, which is one of the Wnt proteins. I think it's like Wnt1. It doesn't matter, but it's a Wnt protein. And what this does is that this induces a different transcription factor called PAX3. So Wnt makes PAX3, and that turns the orange area into the dermomyotome. So it's a combination of the dermatome and the myotome. Both of those are familiar, so you can hopefully guess what those are. If not, we'll get to it in a minute. But what about the sclerotome? What does the sclerotome become? So the sclerotome, that makes the tendons and bones. Sclero meaning hard. So it's specifically going to be like the bones around the neural tube in particular. But the sclerotome is going to code for like bones and tendons. And the dermomyotome is going to code for that, or I guess that's going to develop into skin and muscles. Okay, so there's that. So we've got that bit, and but that's not all, right? So then we have another division that happens in okay, I'm gonna have to redraw that one. This is no good. Okay. So let me see if I can bring back. Oh, my symmetric thing is messed up. Okay, so that should be good enough. But what we've got is that there's two more zones that we got. I really don't like that. Let me turn off symmetry. Okay, so there's two more zones that we get over here. And the orange zone is going to now split into, I guess, three zones actually. So the orange zone, what causes this split to happen? Well, that we've got more transcription factors, of course. So, obviously we need to separate out the dermatome and the myotome because those maps don't look the same. They're pretty different, actually. If you remember from the forearm, from like the upper limb lectures, it's like the dermatome goes like around the hand, whereas the myotome just goes just proximally to distally, just proximal to distal. So these are going to split up during development, and that's going to be what causes those differences. Okay, so let's talk about the proteins that make that happen. So we have Wnt. Basically what happens is that there's a Wnt protein from the dorsal neural tube there, but there's another Wnt protein that's secreted by the this dorsal ectoderm. There's another Wnt protein here. Am I on the right layer? I am not on the right layer. There's another protein that is secreted here. It's another Wnt protein. It doesn't matter which one's which, but the point is that they're different. And they're at the beginning, it's just going to cause them both to make just dermomyotome. But in combination with a BMP, which is stands it so it does. So this is BMP4. You may remember that from the the first part of the lecture. That BMP4 is secreted by the lateral plate mesoderm, which is more on the belly side. Remember, it's on the ventral. I called it belly making protein earlier because it ends up on the ventral side, but actually it stands for bone morphogenic protein, and it's not really making bones here. Doesn't really do any bone making until like part 4 of the lecture, when we talked about like limb development. But BMP is going to come from the lateral plate mesoderm and stimulates the bottom half, which has the bottom half here, I guess it's ventral, the ventral side of the dermomyotome to be different from the back side or the dorsal side. So under the expression of the Wnt protein from the dorsal ectoderm and BMP4 from the ventral, the lateral plate mesoderm, the ventral side of this is going to become this, this is going to become one myotome, and we're going to have another myotome forming in the back. That always happens. Sorry. So there's this red myotome is going to be one myotome, and there's also a brown myotome over here in the back. And then what stays in the middle is going to be the dermatome, that orange space in the middle. So let's talk about the molecular processes that drive these, right? And we've already kind of drawn some of them, but I think it'll help to draw them to kind of make a map of all of them. And so what we start off with, remember, Wnt, right? So that's going to be, I need to leave this. So Wnt is going to be the starter for differentiating the dermomyotome from the sclerotome, and then it makes PAX3, which turns into the dermomyotome. And then what happens is that actually there's another protein that I forgot to draw, but the dorsal neural tube can secrete this protein called NT3, and NT3 that will allow that goes and acts on the orange part, so it doesn't really have as much of an effect on the brown area. It's more acting on the orange area, and that's going to be causing the dermatome part to stay to become the dermatome and not turn into any muscles. Okay, so we start with dermomyotome, and then if we have NT3, then that will turn into the dermatome. And now what if we have something else? What if we have, let's talk about the reds. So actually the browns first. So the brown, if there is the Wnt protein, one of those Wnt proteins, then what that does is that will actually activate a transcription factor called MyF5. My is like a common thing for muscle, so it's like myo, maybe F is like a fiber, myofibril 5 or something, I don't know. But that's going to make our prim axial, or if you watch the noted anatomist video, it's called ep axial myotome. And alternatively, we could also have the other Wnt protein. These are different, remember, but like there's maybe one of them is one asterisk, one of them is 2 asterisks, but we don't really need to know which one's which. And also if there's the BMP4, if we have these two proteins expressed, then this will cause the activation of a different transcription factor, not MyF5, but MyoD, and that induces the formation of the, what do you call it? The ab axial is what she calls it in the video. It's hype axial in the noted anatomist video, but this is the ab axial myotome. So there we go. There's all that's pretty much all the signaling factors. I think that seeing it laid out in a like a map like this for me, at least the process of drawing out the map, is a lot more helpful than seeing the bullet points. But I think this is pretty much what happens. And yeah, so I'll give that a second to soak in. And now let's talk about what the difference is between the brown myotome and the red myotome. So that's going to come in here. This is a graphic that I kind of stole from Noted Anatomist. But if you look, right, so what happens is that the brown side is going to be up here. The red side is going to be down. The brown side is going to become like, oh, whoops, spoiler. But yeah, okay, so the brown side's going to make the back muscles. It's the premaxial myotome is going to make back muscles. According to our lecture, the main ones that you need to know, it's just like the muscles of the back as well as the intercostal muscles and some limb muscles. I'm guessing maybe like the rotator cuff or something, but overall not much. So in contrast, I see I accidentally added 2 layers. Anyway, in contrast, the abaxial is going to generate these guys over here. The abaxial myotome. That's going to make the muscles of the body wall, most of the muscles of the body wall, so like basically the abdominal muscles. So we have external and internal abdominal oblique muscles, can't spell, as well as the transversus abdominis. And most of the limb muscles are going to come from this abaxial side. All right, and yeah, so there's that. And then the dermis is going to come from the orange. So this orange is going to go under the ectoderm, because the ectoderm makes the epidermis, and the orange side is going to make the dermis. Or most of it on the dorsal side anyway. So then what else do we have? Oh, I don't think I pointed out the green side, right? So we talked about the sclerotome earlier, but I don't know if I showed y'all what it makes. So here's the green sclerotome. If you see in this picture, it's making that green vertebra around the spinal cord. So like once development happened, like after a little bit of development, it's initially the somites start looking like this, but once they develop more, these cells are going to spread out, and the green's going to make the vertebra, brown's going to make the back muscles or the premaxial myotome, and the red's going to make the front muscles or the abaxial myotome. Okay, and let me bring back the map. Okay, and I think that's pretty much everything for part 3. So we're going to call it here. And I don't know if I'm going to make a video for part 4, but yeah. Hope that helps. Let me know in the Discord or group me if I'm wrong or if you have any questions, and we can talk about that.